Hello and welcome to Beyond Borders Briefing. In this video, we'll be talking about a remarkable achievement by scientists who have grown an entity that closely resembles an early human embryo, without using sperm, eggs or a womb. This groundbreaking research, published in the journal Nature, was conducted by a team from the Weizmann Institute of Science in Israel. They say their embryo model, made using stem cells, looks like a textbook example of a real 14-day-old embryo. It even released hormones that turned a pregnancy test positive in the lab. The ambition for embryo models is to provide an ethical way of understanding the earliest moments of our lives. The first weeks after a sperm fertilizes an egg is a period of dramatic change, from a collection of indistinct cells to something that eventually becomes recognizable on a baby scan. This crucial time is a major source of miscarriage and birth defects but poorly understood. It's a black box, and that's not a cliché. Our knowledge is very limited, Professor Jacob Hanna, the lead researcher, tells me. Embryo research is legally, ethically, and technically fraught. But there is now a rapidly developing field mimicking natural embryo development. This research is described by the Israeli team as the first, complete embryo model for mimicking all the key structures that emerge in the early embryo. This is really a textbook image of a human day, 14 embryo, Professor Hanna says, which hasn't been done before. Instead of a sperm and egg, the starting material was naive stem cells, reprogrammed to gain the potential to become any type of tissue in the body. Chemicals were then used to coax these stem cells into becoming 4 types of cell found in the earliest stages of the human embryo: epiblast cells, which become the embryo proper, or fetus; trophoblast cells, which become the placenta; hypoblast cells, which become the supportive yolk sac; extraembryonic mesoderm cells. A total of 120 of these cells were mixed in a precise ratio, and then, the scientists step back and watch. About 1% of the mixture began the journey of spontaneously assembling themselves into a structure that resembles, but is not identical to, a human embryo. I give great credit to the cells. You have to bring the right mix and have the right environment, and it just takes off, Professor Hanna says. That's an amazing phenomenon. The embryo models were allowed to grow and develop until they were comparable to an embryo 14 days after fertilization. In many countries, this is the legal cutoff for normal embryo research. Despite the late-night video call, I can hear the passion as Professor Hanna gives me a 3D tour of the exquisitely fine architecture of the embryo model. I can see the trophoblast, which would normally become the placenta, enveloping the embryo. The researchers hope that their embryo model will help them study how embryos communicate with their environment, how they implant in the womb, and how they develop into different organs. They also hope that their work will inspire other scientists to create more advanced and realistic models of human development, as well as animal models for testing drugs and therapies. However, they also acknowledge that their research raises ethical questions and challenges. They stress that their embryo model is not a human being, nor can it ever become one. They also say that they follow strict guidelines and regulations to ensure that their work is done with respect and responsibility. This is a fascinating story, and we'll keep you updated as more discoveries are made. Stay tuned for more science news and analysis on this channel, and don't forget to like, share and subscribe. Thank you for watching.